CPRIT was important, instrumental to my coming to MD Anderson. There's no question about that. I think that I had other offers at the time, very lucrative offers. But when Patrick Hu invited me to come to MD Anderson, and Jim Allison called and said, we're going to make cancer history, I just had to come to MD Anderson. There really was an offer I couldn't refuse, and the CPRIT was really a very big part of that in terms of allowing me to establish my lab very quickly and really explore the use of T-cell therapy in patients with cancer without many restrictions in a very timely manner. And that was most important. Immunotherapy is a way to use the body's own immune system to treat cancer. And unlike other modalities that are commonly available now, which is surgery, radiation therapy, and chemotherapy, including targeted therapy, immunotherapy allows you to treat cancers that are resistant to these other types of treatment because it provides multiple ways of killing cancer. And this can be done using methods that have been established and pioneered by Jim Allison and others, or in a sort of a complementary fashion, you can also try to grow these immune cells outside the body and then give them back to the patient. And that's sort of our form of immune cell therapy or T-cell therapy. We started using T-cell therapy against patients' melanomas because this was sort of a gateway cancer. It's responsive to immune-based regimens, and so it was a logical one in order to develop the program. But now that we've established sort of a recipe or formula, we've come to MD Anderson to sort of expand that into other diseases such as ovarian cancer, breast cancer, colorectal cancer, and diseases that are commonly refractory to conventional chemotherapy and radiation therapy like pancreatic cancer, glioblastomas, and sarcomas. And these are diseases that are amenable to, we believe, this form of T-cell therapy, and not T-cell therapy alone, but in combination with other types of immune therapy, such as immune checkpoint inhibitors. Yeah, I think about a third of the patients have significant responses, and these are patients who failed other types of therapy. This is not ready for mass consumption. It's not something you write a prescription for at this time, but we're really rapidly moving towards a scenario where we can use this modality in more and more patients and in more and more phase 2 and phase 3 clinical trials. I guess there's something to be said for doing science fairs in grade school and high school, and you can have a little plant project, little mice running around in a maze. I actually did a science project, with mice running around in a maze or metabolism in mice, and I think that really got me interested because you could see actual results happening. You have to get that bug, you have to get that experimental bug to say, listen, we do things right, we do things properly, we'll get some sort of an answer. And if it leads to more questions, then that's great. That's an excellent experiment. So we're just doing this on a larger scale now. We're doing experiments that lead to important clinical outcomes. And I think one of the things really is that, you have a chance to make an impression on the field. You have a chance to guide the field in some ways. And it's a unique opportunity. So, it's something that you really would not have a chance to do in any other profession.